Next up, we have Dr. Soichi Shibuya from Great Ormond Street, University College, London, presenting human ex vivo model for congenital diaphragmatic hernia, two-photon polymerization technique to induce mechanical compression to human fetal lung. Dr. Shibuya.
Thank you, Chairman. It's an honor to have this opportunity to present our research at the AXA meeting today. I have nothing to disclose. In congenital diaphragmatic hernia, herniated abdominal organs impose a physical constraint to developing lungs, and they're eventually leading to pulmonary hyperplasia. Pulmonary hyperplasia observed in CDH is characterized by decreased number of air tips and thickened parenchyma. Multiple factors have been hypothesized to be involved in this pathogenesis, but the contribution of the mechanical factor is still unclear. Recently, some investigators published excellent studies whereby they use an excellent vertical compression model and identified some important molecular factors such as FGF10 and periodicity that seem to contribute to the mechanism of the pulmonary hyperplasia. However, there remains much more to explore, especially focusing on the difference of the gene expression between human and the animal models. So we need an efficient experimental model that can be used to investigate the effect of mechanical compression to the human lung development. So together with the collaborators, we established a new 3D bioprinting technique, which is called the two-photon photocross-linking. This method uses a high wavelength laser, which can be generated by two-photon microscope. And if the laser is applied to a chemically modified polyethylene glycol hydrogel, it induces cross-linking and increases the stiffness of the gel. So by applying the laser to a specific area in the hydrogel, so you can create a biocompatible 3D structure in whichever shape you want, as shown in this picture. So we utilize this technique to in the previous research. So we utilize this technique to create a 3D structure like a wall that's confined the mouse fetal lungs to mimic the physical constraint in CDH. And we demonstrate that physical constraint induces a significant decrease, significant reduction in size of the lungs and the number of airway parts in the compressor lungs, of which phenotype reminisces of pulmonary hyperplasia in CDH. So in this study, we try to apply this technique to the human fetal lung samples to investigate the mechanical compression of to investigate the effect of mechanical compression on the human fetal lung development. So we obtained the healthy human fetal lungs from the Human Development Biology Resource of the UK. And we cut the lung pieces into the small pieces and they're embedded in the madrigel and create a 3D structure that's totally confined the lung pieces to generate a physical constraint and incubated them in air-liquid interface culture. These are pictures obtained throughout the experiments. So the lung experiments could survive and keep growing, expanding in size and generating new parts. And we measured surface area to compare the two conditions. And as shown in this slide, so lungs in physical compression were significantly smaller in size compared to the control lungs. We also counted the number of airway tips within the lung specimens and compared it by calculating the rate of newly generated daughter births, and which showed a significantly delayed creation of daughter births in their lungs in compression. So that suggests that branching morphogenesis is disrupted by physical constraint. And finally, we extended the duration of the culture for up to 2 weeks and performed an EDU assay combined with the E-cadherin immunostaining. And the quantification of the EDU-positive epithelial cells showed that lungs under compression had less epithelial cells in active proliferation. So in conclusion, we established a new ex vivo model which can mimic the physical constraint of the CDH. And we demonstrate that physical constraint disrupted the lung growth and the generation of the new daughter births, which may be the target of the pulmonary hyperplasia in CDH. And it's also suggested that disrupted decreased proliferation of fetal cells is a reason. One of the reasons of the branching, impaired branching morphogenesis associated with the physical constraint. This is the end of my presentation. Thank you for listening. I'm happy to take any question.
Thank you. That was a very nice presentation. We will now open it up for questions.
I have one. How did you determine how much compression or how much restriction would model CDH physiologically?
Yeah, it's a good question. So actually, we did an atomic force microscope to measure the stiffness of the gel, and it's actually like 100 hectopascal. So this is a gap of the stiffness of the between the so 3D structure and the normal hydrogel. But it's not actually compression, so it's just a constraint. So we are being careful to describe this effect. So it's not a dynamic compression, so it's a static physical constraint.
Got it. Thank you.
I hesitate to ask this in a room with so many CDH experts, but where do you see this going in the future? Like, what are your next steps?
So in this study, so we are trying to see the effect of mechanical factors. So there have been controversies. So if the mechanical factor is actually important, or genetic mutation is more important in the pathogenesis of the CDH. So we are trying to figure out how much the mechanical factor is contributing to the severeness of their pulmonary hyperplasia. And so what we are trying to do is so identify the pathway which is involved in this pathogenesis of the pulmonary hyperplasia. And if we can find some pathways, pathways involved in this process, so like other presenters showing, so we can do some prenatal therapy. So by stop that pathway, by adding something to their fetus.
Thank you. It looks like we have a question from virtual chat land. Roxanne?
Hi, we have a question from Dr. Pokal. Stretch and strain certainly plays a role in lung development. Thus, in CDH pathology, cellular and molecular mechanisms are disrupted, which take place much earlier than the physical forces on the lung begin during fetal development. So I question the relevance for CDH rescue. How do you justify?
Sorry, I couldn't get the question. Could you please
Sure,
again?
I'll repeat it. Stretch and strain certainly plays a role in lung development. Thus, in CDH pathology, cellular and molecular mechanisms are disrupted, which take place much earlier than the physical forces on the lung begin during fetal development. So I question the relevance for CDH rescue. How do you justify?
To rescue this one. So yeah, it's a good question. And yeah, so we set this experiment so because the physical mechanical compression starts like after around the should granule stage. So, but it's known, so it's very difficult to remove the physical constraint in that area stage. So what we are thinking is to try to find another way. With other, so actually, so physical constraint, it's difficult to remove the physical constraint in the fetus, but thinking about other tissues, so like pancreas, so actually pancreas has a similar development process like branching morphogenesis, but even though pancreas is compressed in the abdomen, so it does have it won't be hyperplastic. So our question is why lungs is only lungs becomes hyperplastic when compression happens. So I guess some particular pathway. So lungs has some particular pathway to how can I say to that's hyperplasia happens. So if we can identify that pathway and make the lung stronger, I mean, even though lung is compressed, so if we can add something to stop that pathway, that leads to the hyperplasia. So we can make a stronger lung. I mean, so even though there is a mechanical compression, so the lung can grow. So if we can make care of that situation, so we can save the children, I think.
Thank you so much, Dr. Shibuya.
Thank you very much.